few minutes లో చేస్తున్నాను. చూడండి సార్.
A very good morning to all. On behalf of Nirmala College of Pharmacy, we welcome you all to the fourth day of AICTE-sponsored Atal FTP on Nano Furtherance of Pharmaceutical Engineering and Industrial Pursuit. Let us start our today's FDP program with a brief summary of yesterday's sessions. In session 7, Dr. Sajal Kumar Jha, Professor, Adams University, gave an outstanding presentation on the application of green synthesis in the development of metallic nanoparticles. Sir gave a detailed description of green nanoparticle synthesis, biosynthesis of nanoparticles, and its application in the pharmaceutical field. In session 8, Dr. Suryakant Swain, Professor, Associate Dean, the Assam Kaziranga University, delivered an excellent presentation on showcasing the QBD framework on self-nanoemulsifying drug delivery system. He explained about quality by design and its importance, need to implement QBD in current scenario. He gave a detailed description on optimization of drug delivery systems, which includes drug delivery development, design of experiments, and computer-assisted data analysis. In session 9, Dr. S. Ravikishore, manager of Izan Pharmaceuticals, delivered a nice presentation on the establishment of analytical procedures for the characterization of nanoformulations. He gave an overview of liposomes, its method of preparation, and its characterization. He also discussed about nanoemulsions and its composition. Today, we will have 3 sessions with 3 eminent persons. Let us start session 10 by inviting Mrs. Supriya Ma'am, Assistant Professor, Nirmala College of Pharmacy, to act as chairperson for this session and to introduce resource person of this session. Thank you to all. Have a great day.
Thank you, Mrs. Srikala ma'am. Good morning to all the delegates and welcome to the fourth day of AICT Atal sponsored online FDP on Nanopharmaceutical Engineering and Industrial Pursuit, organizing by Department of Pharmaceutics, Nirmala College of Pharmacy, Mangalagiri. Myself, Supriya Chatla, Associate Professor, Nirmala College of Pharmacy. It gives me an immense pleasure to introduce Session 10 Resource Person, Dr. N. Bujnaidu, sir. Dr. N. Bujnaidu, currently Professor and HOD in the Department of Pharmaceutics and Biotechnology, and sir is the Director for Siddhartha Pharma Innovation and the Incubation Center. Sir has completed his Masters in Pharmaceutics from Dr. MGR Medical University, Chennai, and has been awarded PhD in Pharmaceutics from Andhra University. Sir worked as Assistant Pharmaceutical R&D Manager, GlaxoSmithKline Pharmaceuticals Limited, India. Sir has been completed his PDF from College of Pharmacy, University of Kentucky, USA. Sir worked as Research Associate, College of Pharmacy, University of Kentucky, USA. Sir also worked as Senior Research Scientist, Yapan Therapeutic Incorporated Company, USA. Sir also served as a Principal Scientist, Yapan Therapeutic Incorporated Company, USA. Sir worked as Professor and Director for PG Studies and the Research, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. And Sir also serving as a Head Formulation R&D, Mitocon Pharma, USA. Sir has received Gold Medal and J.J. Rao Award for his PhD thesis in the year of 2002-3 by Andhra University. Sir has received Best Research Paper Award in Pharmaceutics by IDMA, Mumbai, India. Sir has developed Valaclor, a topical meclorethamine gel which was approved by FDA for the treatment of cutaneous T-cell lymphoma, which is also available commercially in the USA. Sir has 5 research grants approved under various government bodies like DST and the AICT. Sir has received a research grant worth of 4 lakhs on transdermal drug delivery system regulatory requirements in Asian scenario from DST Government of India for conducting an international workshop under Indo-Sri Lanka bilateral program in 2012. Sir has received a research grant worth of 9 lakhs on mouth dissolving film of anti-hypertensive and the anti-migraine drugs for better therapeutic efficiency from AICT. Sir has received a research grant worth of 16 lakhs on microneedle assisted transdermal delivery of anti-migraine and the anti-hypertensive drugs under DST UK India Education Research Initiative Program during 2014-16. Sir has received a research grant worth of 15 lakhs on the development of novel formulation and the analytical technology for some benzodiazepine drugs used for benzodiazepine drugs used in pediatric and the geriatric population under DST, and the Ministry of Sciences and the Higher Education of the Government of Poland Joint Research Program during 2015-17. Sir has received a research grant worth of 30 lakhs on the development and standardization of herbal formulation for the treatment of low back pain as per the regulatory requirements under the India-South Africa Agreement on Science and the Technology Cooperation Program during 2016-19. Sir has Indian and the US patents on the transdermal delivery of the cannabinoids. Sir has published more than 100 research papers in national and the international journals and presented more than 70 research papers in national and the international conferences. Sir has guided 1 PhD scholar and now he is guiding 3 PhD scholars. Sir has guided 70 M.Pharm students and 10 B.Pharm students. Sir is the lifetime member of the Pharmacy Council of India and the APTA. Before inviting the resource person, I would like to inform all the delegates to post the queries in the chat box. At the end of this session, all the queries will be clarified by our resource person, Dr. Bujnaidu sir. Now, I would like to welcome Dr. N. Bujnaidu sir to start the session on nano and the microneedle assisted transdermal delivery of the drugs. Sir, please welcome sir.
Yeah, thank you, Supriya, for your introduction. And I warmly welcome the participants on the online session of the Faculty Development Program organized by the Nirmala College of Pharmacy. So basically today I'm going to talk about the drug delivery aspects through the microneedle concept, device-based drug delivery, especially through the skin, that is the transdermal drug delivery systems. So basically, when it comes to the drug delivery, you need to give a promise to the users, like patients. So drug delivery technologies can increase the likelihood of getting right medicine for the right patient at the right place and at the right time. So overall, the concept is the drug delivery system can make or break a drug molecule. So what it implicates is, if the drug is active, but if it doesn't reaches to the site of action properly, to a right patient, in a right place, in a right time, that doesn't make a blockbuster molecule or a good drug molecule to have a good therapeutic efficacy. So transformation of molecule to a medicine generally comes with an approach of developing a dosage form or into a suitable drug delivery system, so that the drug delivery makes the difference between a great molecule and a great medicine. So assuming the molecule is very effective, either in preclinical studies or in clinical studies, and are assuming that the molecule is not able to reach its site of action in a proper amounts, then the delivery system fails. Even though drug is active, or the drug amounts are not reached to the systemic circulation or to the site of action via a drug delivery system, then the molecule fails. So drug delivery makes a difference between a great molecule to a great medicine. So there are several molecules right now, ranging from small molecules, that is the synthetic molecules. You have peptides, you have proteins, you have monoclonal antibodies and oligonucleotides, or broadly small and large molecules. So different molecular formats present unique delivery changes or challenges. So what happens is here, when you come to the small molecular approach, delivering the molecules, so you have a problem with solubility or oral bioavailability. When it comes to the peptides, you have a problem with solubility and stability. And when it proteins, it is stability and aggregation. And when it comes to the MAPs, monoclonal antibodies, you have a problem with solubility and viscosity because of the high amount of the monoclonal dose and aggregation problem. And oligonucleotides, specific targeting and stability aspects. So what happens is, diversity of disease targets lead to diversity of molecular formats. So from a small molecule to now vaccine delivery now is the major topic or the major issue around the globe, especially when it comes to the pandemic situation, the corona vaccine. So the corona vaccine is now delivered through the intramuscular route in injection form or in liquid form. So you never know. So but there are problem with the logistics, that you need to store that at 8 degrees centigrade, and you have to use it within a period of time when you bring it to the room temperature. So you have a problem with the stability issues. All these kind of issues can be solved. You can deliver the vaccine in a suitable delivery system like microneedle approach. So already there were several papers published on the concept of delivering the vaccine using the microneedles. So when you talk about the advancements of the drug delivery, whether it is in academic purpose or in an industry purpose research areas, so you have the first technologies like simple elixirs and simple solutions, and then the second technology, tablets or capsules or spansules or the transdermal patches, and in the third technology you have the monoclonal antibody injectables, and the emerging new technologies like the stealth-based liposomes or nanoparticles or microneedle drug delivery technology. So day by day or gradually the technologies or many technologies are under the research, so that the drug can be delivered to a right patient in a right way in a right time. So with the new advancement of these drug delivery technologies, now the concept is that it's not necessarily the formulation guys or somebody can involve with the developing a suitable drug delivery system, especially when it comes to the device-based drug delivery systems. So several people, several people involved in it, especially it's not necessarily the pharmaceutical guys. So people who have knowledge in the biology, bioinformatics, chemistry, biochemistry, engineering, clinical sciences. So it's a complex phenomena, and basically it is a flexible, interdisciplinary approach, so that a good platform of the drug delivery system can be developed, and that platform can be used for any purpose or delivering any type of the molecule. So that's the concept now is going on in both in the academic sector and in the industrial research, so that to deliver the molecules. So there are several challenges when you want to deliver the molecules, especially. So when it comes to the oral drug delivery, you have a problem with the class 2 and class 4 molecules. And when you want to develop an injectable delivery system, the high dose of proteins, monoclonal antibodies, peptides, we have a problem with the viscosity and aggregation, as I mentioned earlier. Whereas when it comes to the parenteral sustained delivery of the proteins and peptides, you have a conjugation or the formulation or the default issues or the developing the molecules or conjugated, polymer conjugated regular system, and alternative routes most preferably for delivering the proteins and peptides, including or even also the small molecules, pulmonary, nasal, oral, buccal, and transdermal. So today we are going to talk in much detail about the transdermal drug delivery systems, and also you have the targeted delivery system, and with the nanoparticle technology and combining it with the transdermal route of delivery, you can have a targeted delivery system in both ways, and delivering of the emerging modalities, now the siRNA and stem cells. As I mentioned, the COVID vaccine, it's an mRNA-based vaccines, the Pfizer product, that is Moderna Pfizer vaccine. So broadly, today I'm going to talk about the nano microneedle assisted transdermal delivery of the drugs. So what happens? The drugs through the skin. So how you can deliver the drugs to the skin? So basically, to date, you have a patch, transdermal patch that you put onto the skin, and the drug goes into the systemic circulation and then to the therapeutic site as a way of an injectable dosage form. So basically, when you deliver the drug through the transdermal route, you may anticipate parenteral route of administration activity with that of the molecule. So which one is more preferable? Types of tablets or patches or injections? Okay, individually they have several advantages and disadvantages. So some say they have patient friendly, some say there is no complication, or some say injectable pain, but depending on the need or of the hour, the patient may be given an injectable formulation, the patient may be given patches, or the patient may be the tablets. So each and every route of administration, each and every drug delivery system has its own pros and cons. So basically, when you develop a transdermal drug delivery system in the earlier days, the transdermal drug delivery system means it comes under the controlled drug delivery concept, so that you have a therapeutic levels maintained within the therapeutic window over a period of 8 hours to even up to 1 week also. So the hormonal patches were available in the market that you can wear them up to the 1 week, so that the drug will be released continuously. So over a period or 1 day to even 7 days, also the drug can be delivered through the transdermal drug delivery system. But the advantage with the transdermal drug delivery system is that the concept is that you are applying the drug delivery system external to the body. So that means you have every chance that you can withdraw the dose, and which is not possible with the parenteral route of administration or oral route of administration or any other route of administration especially. So the topical or transdermal drug delivery has an advantage of dose withdrawal at your convenience. So, which is a great concept that I think it plays an important role in the future for delivering the molecules. So basically, the skin functions for several purposes. It is a barrier purpose to prevent the toxic substances entering into your blood or protect from the UV light, or it's a surveillance action, continuous turnover, and others like vitamin D synthesis. So the skin, it's a multiple functions, the organ, and basically it is the largest organ of the body, human body. So it basically comprises epidermis and dermal layers, and as a part of the epidermis, the viable or visible layer on the top of the skin is the stratum corneum, which is about 10 to 20 microns thickness, and then under the stratum corneum you have an epidermis which is up to 50 to 100 micron thickness, and under the epidermis you have around 1000 to 12 microns dermal layer, and under the dermal layer you have a capillary network where the drug goes directly into the systemic circulation. So basically, when you talk about the skin structure in terms of transdermal drug delivery, the layer you talk more about is the stratum corneum, because this is the outermost layer which protects the body, and also at the same time which is a hurdle to deliver the drugs. So basically, it is because that the lipid composition of the stratum corneum layer which prevents the drugs to enter into the skin and into the systemic circulation. So bypassing the stratum corneum layer is a major goal, especially when you deliver even from small to large molecules. So small molecule means it not necessarily all the molecules will permeate through the skin. So the molecule which are having a good solubility and good log P value, optimized values, can only be permeated through the skin. So that means, ideally speaking, the molecule should have both lipophilic and hydrophilic properties, can easily permeate. The molecule is too lipophilic, also cannot permeate through the stratum corneum, because the stratum corneum is lipid in nature. So the lipid molecule stays in the lipid layers, and the drug will not cross the epidermal or the dermal layers and enter into systemic circulation. Whereas vice versa, when it comes to the hydrophilic molecules, because of the not optimized log P value, so the molecule doesn't permeate through the stratum corneum, and molecules enter into systemic circulation. So basically, the ideal molecule should have both the hydrophilic nature and lipophilic nature. Then only the molecule is suitable to delivering the transdermal drug delivery system. So older days, people used to say, even still we read in the textbooks, that the ideal molecule for delivering the drug through the transdermal route is its restriction of the molecular weight and solubility, or its lipophilicity, or some other structural aspects, physicochemical aspects. But with the invention of the new techniques that we discuss in the coming slides, so any drug can be now delivered through the skin, whether it is a small molecule or large molecule like proteins and peptides or vaccines. Now any drug can be delivered through the skin in a good suitable amount, so that have a therapeutic efficacy. So basically, when the needle, basically what happens is when you inject, you get a pinch, because it touches the nerve endings, the capillary endings, where you get the blood circulation and you get pain. But whereas when it comes to the micro needles, is that you cannot, or you can minimize the pinching of the needle with that of the capillaries, and you don't feel any pain. So that means both pediatric or geriatric populations, or any kind of population, can be easily administered with the microneedle delivery systems of all kind of the drug molecules here. So whenever you talk about any route of delivery or any drug delivery system, at the end of the day, you have to think about the commercialization aspect. So basically, if any company looks in that aspect, when they develop any drug delivery platform, you make sure that it is commercially viable. Otherwise, you spend millions of dollars on that, and then you get nothing from it, and it's a big loss to the pharmaceutical companies or even to the academic research also. So the transdermal over a period of the 30 years or the 30 to 30 years, there were several products were marketed so far, basically on the concept of transdermal delivery. Especially they are the transdermal patches. So they are either the reservoir patches, or they are the adhesive drug-in adhesive patches type. So starting from the beginning, so far you have so many patches that were available in the market. Even in India also they are available in the market. So even the talk we are now going to discuss about the microneedle. Also, there is one product is approved by the FDA, and I think that this product was called back because of the some issues. I think it's no longer available, but several companies are working on the microneedle approaches, delivering the drugs, and they are nearly to filing an NDA on that. On the concept of the microneedle delivery. So when you talk about the revenue sources, so you got a good market in the sense of the delivering a transdermal product. So people can do research based on the transdermal drug delivery platform, and they can easily commercialize the product. So that is why many companies working on the transdermal drug delivery systems based on the microneedle delivery. So as I mentioned earlier, does all the drugs permeate through the skin? It's a question mark. So, okay, if they permeate, okay, they may permeate, but that doesn't mean that the amount that is permeating through the skin is not good enough to have a therapeutic efficacy. So that is the issue here. So this is the reason, even if you see so many papers, thousands of papers published on delivering the drugs through the transdermal drug delivery, either through any approach, whether it is a transdermal patches or some kind of the alternative delivery that enhances the drug delivery through the skin. As you mentioned here, some active vehicle interactions. You have a prodrug concept, you have a chemical permeation enhancer concept, and ion pairing and eutectic systems, and vesicle-based systems like liposomes, microemulsions, lipid nanoparticles, high velocity particles, and you have stratum corneum modified, so hydration and chemical enhancers, and stratum corneum bypassed, especially the microneedle or ablation technology or follicular drug delivery. Even the some time back, 15 years back, the Pfizer company also investigated on the delivering of the drugs through the hair follicular route. So you have a potential opening there, so drug can be delivered through the hair follicles. And also the electrically assisted, especially the ionophoric electrophoration. So several concepts were introduced to enhance the drug delivery. So the basic bottom line is here, as I mentioned, you may permeate the drug through the skin, but the end of the day, you need to make sure that the amount of the drug permeated through the skin into reaching into systemic circulation is good enough to have a therapeutic effect. So that's the concept here. So with that, I think we covered almost the concept of the transdermal drug delivery. And now the continuation of the slide mostly will talk about the microneedle based drug delivery systems, how these microneedles will be helpful in delivering the drugs through the transdermal route of application. So as I mentioned earlier, you have a passive DIA drug-in adhesive technology. So you have a molecule polarity neutral, molecular weight 500, melting point 200, or drug dose is a limited dose. So there is a problem with the transdermal delivery. In older days, when especially you use the transdermal patches. So high doses drugs cannot be delivered through the transdermal route because it is very difficult to permeate such a high amounts sometimes. But even small amounts also may not good enough to have the therapeutic efficacy. So when it comes to the gels, thermal technique, ionophores, sonophores, or electroporation, so the microneedles, the issue is that irrespective of its lipophilicity, irrespective of its hydrophilicity, or bottom line, irrespective of its polarity, all the molecules can be delivered through the skin using the microneedle technique. And as I mentioned, there is no restriction with the molecular weight or melting point, which replicates back to the reflects back to the polarity of the molecule. And dose, yes, dose is maybe a problem. So high doses may or may not can be administered using the microneedle technology. But definitely that can be solvable using the different microneedle approaches, different types of the microneedles that I will briefly explain in the coming slide. So what is it to do with the nanotechnology? Because I am here talking mostly about the micro, and what it is do with the nanotechnology. And these are the different microneedles that we tried in our lab under the DST UKD grant of the microneedle assisted transdermal delivery of the anti-migraine and anti-hypertensive drugs. And I'm going to talk about the work we done in our lab by taking a consideration of the one small molecule and one large molecule. Under the small molecule category, I will talk about the sumatriptan, and under the large molecule concept, I will talk about the insulin. So what it is exactly the approach when you talk about the delivering the drugs through the microneedles? So basically, you have four types of approaches. The first one is the coat and poke. So that means the microneedle tip is coated with the drug uniformly coated, and then you insert the microneedle patch onto the skin, and the drug will be dissolved and released into systemic circulation. So several papers were also published from this concept. And the second concept is poke and flow. So this is the concept that you can use to deliver, because here you are using a hollow type of microneedles. You see here there is a small hole, or a continuous flow can be achieved through this kind of the hollow microneedles, and over that you put a drug reservoir, and the drug can be delivered continuously, which will give a kickback like an IV infusion. But the issue is here, which is a patient friendly. So when compared to the catheter inserted to your into your veins, and which is not mostly the patient friendly. So whereas with the microneedles, a tiny microneedle patch is applied onto the skin. Over that you have a drug reservoir, and from the drug reservoir, the drug is continuously available into systemic circulation. So you have an advantage with that. And I think with the poke and flow approach, I think you can minimize the problem with administering the high dose of the drugs. And the third category, the poke and release category, where you have a dissolvable microneedle. This is where the concept of the nanotechnology arises here. So this is where the nanotechnology is merged with the microneedle technology. So the microneedles made with the nanoparticles or other vehicles like liposomes, so that you make a dissolvable microneedle patch, an array, microneedle array, that we are now doing in our lab right now. So, but it is too early for us to talk about the data. So that's why I give a basic concept of the microneedle technology in this presentation. Hopefully in future I may share the data that is available in future. So the nanoparticles or the vesicles can be mixed with the polymer mixture that you form to make the microneedles there, and then you can make the combination of the nanotechnology and the microneedle technology, and you can deliver the drugs through the microneedle technology. for the purposes of targeted delivery. I will talk in the coming slide. And also you have a poke and patch. So you make the microneedle patch arrays applied onto the skin so that you get a small pores onto the stratum corneum, so that the pores make viability of delivering the drugs or viability of permeating the drug through the skin in good amounts without having any issue, so that the therapeutically effective drug concentration can be reached into the systemic circulation with this approach. So what basically is, as in the nanotechnology, I'm just here quoting 2 papers, but there are several others around the world, especially in the academic sector. People are working in combining the nanomedicine technology or the nano technology, delivering the drugs through the transdermal route of application. So basically what it is here is you have microneedles that may come along with the liposomes and the polymeric nanocarriers and the iron nanoparticles or nanosuspensions or mesoporous silica nanoparticles. So for therapeutic purposes, different purposes. So you can have a targeted delivery, especially when it comes to the cancer, the therapy, chemotherapy, immunotherapy, photodermal therapy, and photodynamic therapy, and the therapeutic proteins. And you can also deliver the proteins and peptides also there, even the monoclonal antibodies, including the hormones and enzymes. And you can also use this technology for the gene therapy. The siRNA and the DNA can be easily delivered using this the microneedle technology. And most importantly, the vaccine delivery. I think in future you may see a product delivering the vaccine using the microneedle technology in future. So I think several companies are working in that with that concept. So because it is very easy to have when it comes to the logistics purposes. So transferring the vaccine from the site of manufacturing to the where the drug, the vaccines are applied. So using this microneedle technology, you just put a patch, microneedle patch onto the skin, and which is stable at room temperature. So which you can minimize the expenditure that you do while transporting and storing of the vaccines. So hopefully we may see some a vaccine delivery technology based on the vaccine delivery system based on the microneedle technology. So from this here, I will talk more about the delivering the drugs as a basic concept of the molecules delivered through the microneedle approaches. So what we did is in our lab in 2014, I think we got this grant in 2014, and up to 2017 or 18 also we continued with the several PhD and MPharm scholars worked on this project. And several MPharm people worked with the delivering the microneedle, and we have published so many science-indexed papers. So on this, so basically what we did is we took the commercially available stainless steel, medical grade stainless steel microneedles, which are manufactured by the Admin Med with the different lengths in it. So you have 0.6 mm, 0.9 mm, 1.2 and 1.5. So as the length increases, so the number of the microneedle decreases, because the area of the array is the same. So when the needle is increases, naturally dimensions also changes. So when the dimension changes, then the number also reduces drastically with the increasing the length of the microneedles here. So as I mentioned earlier, so the microneedles may not touch with the capillaries, so you don't feel any pain, and potentially they are painless administration of the drugs or delivering the drugs through the skin. So what happens is when it comes to the delivering the drugs, especially molecules with the class of the triptans, which are anti-migraine drugs. So the basic concept is presently these molecules are available as immediate release tablets, and also some are available as the mouth dissolving tablets actually. So the bottom line with the mouth dissolving is that to enhance the oral bioavailability, that you encounter less than 50% with the oral route of administration.
போடல சார் மறந்துட்டேன் இதோ போடுறேன் இதோ வரேன் இல்ல
I think somebody has to mute, it seems. So
யாரோ அன்மியூட் பண்ணிருக்காங்க
okay, so the concept is that if you administer 25 milligram or 50 milligram of sumatriptan, so less than 50% of the drug is entering into systemic circulation, and that is having the therapeutic efficacy. So that means if you administer 25 milligram of sumatriptan, only 12.5 milligram is reaching the systemic circulation or less than of that. So that means you are administering an amount that is needed more than the amount that is needed. So that means the side effects that you may see with these kind of the molecules is not because of the drug itself. It may be because of the dose that you are administering. So you are administering almost 50% more dose through the oral route of administration. So the basic concept is here. So if there is any alternative route of delivery, we can deliver the drug, especially the amount that is needed, that is 12.5 milligram, then you may see the cost of the therapy is reduced, and more effectively the drug reaches the systemic circulation and having therapeutic efficacy with less administered dose, and cost of the therapy also may reduces. So what's the issue then? Can we deliver these drugs or not? The problem is these drugs are hydrophilic in nature, so many of them are salts and they are in hydrophilic, and they don't have a good log P values that can bypass the stratum corneum. So the microneedle technology, what we used, they can bypass the stratum corneum and deliver these triptans to the systemic circulation. So the aim and objectives of the study that we carried out is initially you need to have a good quantification method. So whenever you talk about the transdermal drug delivery system, so you need to have a good sensitive analytical methodology. So so far the best analytical hand-on methodology that is available in our labs is the best one is the HPLC methodology. So we developed an HPLC method for it, because you have to make sure the sample volume that you get from the transdermal experiments is few microliters only. So you need to have a good sensitive analytical methodology, and selection of the suitable donor vehicle that you keep on the skin along with the drug, so that that will give a good saturation amount that is available throughout your experiment period. So for that we did some solubility studies to select a suitable donor vehicle, and then we did the in vitro permeation, skin permeation studies using the pig ear skin as a model. So there are several researchers use several skin models. So if it is if you get the human cadaver skin, it is fine. So which is nothing but a human skin and alternative to the human skin. But whereas when it comes to the human cadaver skin is not available, especially when in countries like in India, because you don't have a cooperative tissue network that is there in USA and other countries, where a human tissue can be transported within a day from one place to another place throughout the country, especially in USA. So you get a human cadaver skin there. But here in India, it is very difficult to get the human skin unless until it is comes through through the government resources. So the alternative model skin that you use is the abdominal skin, rat or mouse abdominal skin, that is the common, and you can easily use the pig ear skin. So we selected the pig ear skin because the lipid components of the pig ear skin are almost the same as that of the human stratum corneum lipid content. So, which gives the near comparison to the human skin data. So that is why we selected the pig ear skin as a model of the membrane here in our studies. And skin perforation by the microneedles and the rollers, and visualization or histological examination of the microneedle skin, how the needles making the microconduits or micropores when you apply the microneedles onto the skin using these histological sections, and evaluation of the skin permeation data, how much drug is available, how much drug is permeated, whether the amount of the drug that is permeated is having any clinical relevance from the in vitro flux data. So basically, these are the HPLC method conditions. We used the ammonium acetate and methanol combination as mobile phase, and we got a good sensitive peak at around 3.61 minutes with an injection volume of 10 microliters. So the chromatograph and the purity curve and the UV spectrum using the UV PDA detector data, this is. And the method we got is very sensitive, which is able to detect the levels, even the amount permeated through the skin also. So assuming that not even milligrams is available, not even micrograms is available through the skin. So sometimes you may have nanograms also. So that's why your method needs to be very sensitive to quantify even the nanogram range of the drugs in the receptor fluids. So when it comes to the solubility studies to select a suitable vehicle, we got two vehicles. One is the polyethylene glycol and saline, and propylene glycol and saline at different combinations, 90:10, 80:20, and 70:30. When it comes to the 70:30 ratios, we got the good solubility for both the vehicles. So based on the PEG and the PEG. So PEG we used here is the PEG 400, which is liquid in nature. So, but we selected based on this data and the skin compatibility and the solubility, PG saline 70-30% saturated drug solution was used for doing the transdermal permeation studies. And we did these studies using the Frange diffusion cells, where you keep the skin layers that now I am showing here. The microneedle poked skins layers, they were dyed and to clearly visualize the micropores onto the skin layers. So you have several microneedle lengths that were applied. So when compared to the control skin, you can see very clearly the micropores that are formed over the skin. So as I mentioned in the previous slide, you see the microneedles are more with the 0.6 mm microneedle length when compared to the 0.9, when compared to the 1.2, and when compared to the 1.5. But the length is more, the size of the microneedle is more, and the size of the micropore that is formed over the skin is also more. You can see very clearly there with these structures. So these are the derma rollers that are commercially available. You make the skin that pass the derma roller over skin so that you may get. But the disadvantage with the derma roller is that the microconduits or micropores formed not in a fashioned way, in a non-fashioned way, irregular way. So you don't have a control over the formation of the micropores over the skin. But we did this study when we wrote the grant proposal, because whenever you write a grant proposal, basically in the grant proposal you need to give some previous data that conveying the grant authorities to say that we already exercised in conducting this grant, or to have this technology that can be implemented in our grant. So initially we got these derma rollers from the market, and we did some studies, and the data was submitted to the DST, and then ultimately they have sanctioned this project. So these are the histological sections of the skin where you have a control. Please, can somebody move you?
কার আনমিউট করা আছে পুরো জায়গা থেকে এত
Okay, control skin, if you see very clearly, the skin is intact, or the superficial layer, stratum corneum especially, is intact along with the epidermal and dermal layer. And but when you apply the microneedles, if you see, there were micropores formed, and the microforms formed. The length and the diameter increases with increasing the length. This is the 0.6 mm microneedle applied, and this is 1.2. And when they expose it to the same imaging technology and with the same magnification, you see that the 1.2 micropore is very visibly very clear that it is the width is higher, length is higher when compared to the 0.6 there. And the further the length and width of the micropores formed was calculated using the TOPU software. You fit these histological section images to this TOPU software, where they can expose it to the same magnification, and you can calculate the length. So from the length calculated, you see that with the 0.6 mm microneedle technology, you got around 352 microns depth, whereas with 1.2 you got around 660. So almost very clearly, of or 50 to 60% of the length of the microneedle is making penetrating into the layers of the skin. So don't think that the entire length of the microneedle goes through the skin, because skin is like a puffy like structure. So what happens is that it prevents the insertion of the microneedle. The bottom line here is that 50 to 60% of the microneedle length only will be pierced into the skin. So that means that the technology is safe, and not necessarily it will touches the capillaries. Even if you use the 1.5 mm microneedle, that reaches where the systemic circulation, I mean capillary zone, and you don't feel any pain with this microneedle technology. And there are no disruption of the capillary zone, so that you may not get any bleeding or at all. So this is the beauty of the microneedle technology. And these are the in vitro skin permeation profiles over a period of 48 hours using the different technologies or different microneedle lengths or derma rollers. So you see the PASU, the bottom line, you have as the length of the microneedle is increasing, the permeation of the drug is increasing. And let us see the data that you obtained from this permeation profiles in this table. So you have a flux that is in the expressed in nanomoles, or you can express in micromoles or millimoles also. It's not an issue to a specific area, to a specific period of time. And you have lag time. The lag time is the time that is where you get the straight permeation profile time. Where you have a permeation is started in a steady state fashion way. So, and then you got the permeability coefficient, and you got the diffusion coefficient based on the solubility data. And you see that as the length of the microneedle increased, the flux is also increased, and the lag time is drastically decreased. So the lesser the lag times, the quicker the drug permeating through the skin and entering into systemic circulation, so that you can have a quick onset of action and quick and an immediate therapeutic effect, especially as like in the intramuscular or intravenous route of administration of the drugs. So the microneedle technology, hopefully in future, may be an alternative to intramuscular, especially a route of delivery, keeping in view of the delivering the large molecules, especially vaccines. So if you see the flux that you got with the passive delivery, that is without applying the microneedles, you got around 255 nanomoles per centimeter square per hour. And here is n is equal to 3. And whereas with the 1.5 mm application of the microneedle, which is almost the 30 times more when it comes to the flux, and almost around 20 times less of the lag time, and the permeability coefficient is also drastically increased to around 20 times, and the diffusion coefficient is also increased around 18 times. So the bottom line here is the microneedles effectively and significantly enhanced the delivery of sumatriptan by increasing the needle length. So a good amount of the sumatriptan is reached to the systemic circulation. So now, as I mentioned earlier slides, okay, you may deliver the drug, but you at the end of the day, you make sure that the amount that is delivered into the systemic circulation is good enough to have a therapeutic efficacy. That is the concept of industrial R&D. So anybody can deliver the drug to the skin. That's not a point here. The point is that whether the drug is good enough or not, the amount is good enough or not to have a therapeutic efficacy. So let us see with the 1.5 mm microneedle flux value that fit into the calculation and see whether it is therapeutically effective or not in the coming slides. So before that, we made some correlations with the height and the thickness of the skin layer and the length of the microneedle and the amount permeated. We made some scaling analysis to predict the amount that is permeated between the different microneedles. So you see that with the microneedles, we got one is 0.6 and 0.9. And we don't have 0.7 or 0.8 or 0.5 or something like that. So from the 9, it is 1.2. We don't have 1 mm microneedle. So with the scaling analysis data, using the surface area of the skin that you used or the length of the microneedles, so one can easily predict the flux values. So by not doing the transdermal permeation experiment using this mathematical or the simulation analysis, one can predict the different microneedle ranges flux values. You see here a good correlation with increasing the length of the microneedle increased the transdermal flux in a linear fashion way. One can easily predict different needle lengths between 0.6 and 0.9, 0.9 and 1.2, and 1.2 to 1.5. Any needle length can be done. But this doesn't mean that you may get the same data for all the molecules. So the flux data vary from molecule to molecule based on its physicochemical properties. But when it comes to this sumatriptan molecule, you got a good correlation. So one can easily estimate the different lengths of the microneedles, the flux values for the sumatriptan. So in the discussed in the previous slide, the flux can be calculated using the area of the and then the steady state concentration and the clearance value, the human clearance value for the sumatriptan. The CSS is the 72 nanogram. That is the steady state that you take consideration, especially when you want to develop the sustained delivery. And the required flux for 1 centimeter square area patch is the 17,000 nanomoles. But whereas with the 1.5 mm microneedle treatment using, remember, here we have not applied any dosage form. It is just a basic study that you apply a saturated solution of propylene glycol and saline of this sumatriptan, and where you got a flux of 6,754 nanomoles with the 1.5 microneedle. That is per centimeter square area. And the surface area, overall area of the skin that is needed for the application of the 1.5 mm is the 2.5 centimeter square area. So the area in the France cells we used is only around 1.77 square centimeter. So the circle that you get with the orifice diameter of the France cells we used is the 1.77 centimeter square only. So that is why you need the mathematical calculations to see how much area is needed. Okay, now you got good amounts permeated sumatriptan. So if you make a transdermal microneedle array patch of 2.5 centimeters square where you get a good amount of the drug is available over the donor side, and you can definitely deliver the sumatriptan. But in practical, or in making a dosage form, or in making this technology into the commercialization, then you need to optimize the exact scenario that we see in the experimental section, where you used the saturated solution, which may not feasible when it comes to the product development, especially to area of 2.5 centimeters square. Okay, you may enhance the area, it is not an issue. But the bottom line here is, from this studies, a significant amount of sumatriptan can be permeated through the skin using the microneedle technology, and these good amounts can be sufficient enough to have a therapeutic efficacy, keeping in view of the 50% oral bioavailability that you see with the oral route of administration. So only 50 or less than 50% is available. So that means around 12.5 milligram is assuming is available. So that 12.5 milligram can easily delivered by making a dissolvable microneedle technology, or a poke and patch, where you apply a transdermal patch of sumatriptan, and you on the poked skin you apply patch, and the drug will be continuously released in a sustained release fashion way or controlled release fashion way. So bottom line is here, the in vitro permeation studies demonstrated the microneedle pretreatment can significantly enhance the sumatriptan permeation, and the enhancement is depending on the length of the microneedle, and the superior with the length of the needle increases, the permeation is increased. And when compared to micro rollers, the needles have the more permeation. And with this, to this sumatriptan section, I can easily say that using the microneedle technology, sumatriptan can be delivered through the skin in a significant amounts that are having the therapeutic efficacy. So now let us see using the large molecule. So the large molecules, you have a problem like the burden of daily injections when it comes to the insulin, because insulin you need to take one or twice a day injections depending on your condition, and the injection is sometimes self-administration at home, or sometimes if the patient is in coma, they need to be given in the hospitalized conditions, and the patient may be compliant or may not compliant, and there are some issues like physiological stress, inconvenience, and cost, and localized deposition of the insulin in the fatty layers, where the insulin will not permeate or reaches the systemic circulation. So there are several issues, especially when you give insulin in an injectable or formulation. So given the very low diffusivity of the insulin in the stratum corneum, and owing it to the large molecular weight as like other proteins and peptides, so you need to alleviate the stratum corneum effect in permeating the insulin. So insulin is a biggest disaster in the world, and more than 387 million people worldwide are diabetic in nature. So I think in each and every household, you may see one or two patients, diabetic patients, regularly in our households. So in the same way, we carried out the experiment with the sumatriptan. We also did with the insulin here. And we also developed a good analytical method, sensitive analytical method, that is able to detect the low levels of the insulin using the 0.2% formic acid and acetonitrile combination using the inert cell ODS C18 column. And the diluent is the sodium chloride, that is saline, 0.9% saline, and the retention time is around 6.8 minutes. So again, here using this PDA technology, you can say very clearly the percent purity or the identification of the insulin, and using this technology, and we used this method for further quantification of the insulin in using these transdermal permeation studies. Here also, if you see, we conducted the studies up to 6 hours here using the marketed insulin preparations, pouring the significant amount of the volume over the skin and see the different needle lengths. And you can see from very clearly from the profiles, formation profiles, very clearly the needle length increased. The permeation of the insulin also increased, and we also calculated the cumulative amount permeated here. Because if you see from with the passive, you got the 452 microgram per centimeter square, whereas with the 1.5 you got around the 789 micro, so almost twice amount, around closer to twice amount. And the lag time is also drastically decreased with the increasing the needle length. But the diffusion coefficient also is, but when it comes to the permeability coefficient, very 2 times, around 2 times, so not as like 10 times or 20 times when it comes to the small molecule. So the diffusibility, permeability, when clearly say the low diffusibility, low permeability of the insulin molecule that you got with the delivering the drugs through the transdermal route of administration, especially the protein and peptide molecules. So we also done using the same scaling technologies to correlate the amount permeated with the microneedle length and area. And here also you see that a good correlation is obtained so that you can predict the insulin permeation. And actually, we did this work in collaboration with the University of Loughborough, that DAS laboratory, University of Loughborough, UK. Actually, one of my PG student visited that lab and he stayed for 1 month there. And we also visited, myself is also visited there. And we did some studies there. And this numerical simulation study we did there in the Dr. Das lab, where they developed a numerical simulation data based on the skin sections and then the diffusivity of the molecule, the permeability of the molecule, or the amount of the molecule, and the time you applied. What they did is they using the COMSOL software, they have predicted the in vitro permeation. So what we done is we compared we simulated the data that is obtained with the simulation and the data that is applied with the in vitro skin permeation. We just compared, and the similarity factor is found to be greater than or equal to 50, assuming that saying that with the simulation also one can use to predict the data, especially when you have the histological sections of the skin. So that you can predict the in vitro permeation data. So what all this makes is that as a QBD approach, or whatever you call, so these simulation or mathematical approaches will reduce the time of the experiments to get the realistic data, so that the formulation scientist will get an idea with the minimum data on hand, so that the product development can be faster. So that's the basic concept of the QBD. I think yesterday Dr. Swain gave a good explanation about the QBD approaches with the self-emulsifying nanodrug delivery systems. So the bottom line here is that okay, insulin is permeated, but as I said, insulin amount that is permeated may or may not enough to reduce the glucose levels. So that we need to see we did in the some studies in the rats, but I don't have the data here. I'm not going to present the data. Similarly, the scientists from the North Carolina University, they developed a microneedle array patch with the insulin, where they have the glucose sensing enzymes. So whenever the glucose levels are high, this microneedle patch, it triggers the release of the insulin into the systemic circulation. So that means if you have a chronic diabetes, you can just put this microneedle patch onto the skin over a period of time. And if this device monitors your glucose levels using these glucose sensing enzymes that are there in that particular device, which are in the microneedle. And at the same time, the insulin is also triggered to release from the microneedles. So I think this technology right now in the clinical phase, I think they have almost done with the phase 1. They are now under the phase 2 and phase 3. and hopefully we may see this technology in future that is available in market or commercialized successfully. So not only this insulin, several vaccines, as I said, are several large molecules, even monoclonal antibodies, or any kind of the protein or peptides, or anything can be delivered by suitably entrapping the nanotechnology based large molecules into the microneedle technology, a device-based technology, so you can successfully deliver the drugs. I hope that within the decade we may see several products into the market using this technology. And we are very much thankful to the Siddhartha Academy for providing the necessary facilities to carry out this work, and Milan Laboratory for providing the sumatriptan granules, and NRI Medical College and our Siddhartha Penamneni Siddhartha Institute of Medical Sciences faculty to have these skin sections there. And most importantly, Dr. Das, who is our collaborator under this DST UKD project. And with this, I take very proudly, I can say, this is the first microneedle funded project in India by Government of India. So we got this in 2014, and we have successfully completed, and we published more than 6 science research papers. And it's not only that, we got a good technology transfer from India to UK and UK to India. And the students from the University of Loughborough visited our college, and students from our college visited the UK, and faculty also exchanged, and we also organized several international and workshops and seminars under this program. And we are very much thankful to the Department of Science and Technology, International Cooperation Division, and Government of India, and the British Council of India through the UK India Education Research Initiative Program. We got this project sanctioned, and we were very fruitful with this case. Thank you very much. And these are the students who worked under this project, especially Anusha, who is a PhD scholar. Right now she is submitting, going to submit microneedle technology-based PhD thesis to the Krishna University. And the blue shirt guy is Chandra Teja, who published as a first other 6 science-indexed papers from our lab by doing the MPharm project work. And he also visited the UK, and later on he applied for the Lady Tata Fellowship, and he got that fellowship, and he also completed PhD. He joined PhD in the BITS Pilani Hyderabad campus, and he also submitted his thesis. And the another girl is Ashraf. She also worked on the, several people worked with the several molecules, delivering the molecules through the microneedle technology. And without their support, I may not have finished this project. And I am very much thankful to them also. And once management, Sister Maria and Alfonso, and the young and dynamic principal, Dr. Rahman, and the convener or organizer of the online FTP program, Dr. Reddy, for giving me these opportunities. Now I am open for the question session. Thank you.
Thank you, sir. Thank you for your wonderful and valuable presentation, sir. Sir gave the depth information on microneedle assisted transdermal delivery of the drugs. Sir, thank you for your presentation, sir. Sir, now I request you to address the queries raised by our delegates, sir.
Sure.
Can I ask the question, sir?
It's fine. If there are no questions right now, they can contact me.
Sir, we have 2 questions, sir, from delegates. Can I ask, sir?
Sure.
Sir, here is the first question, sir. How the dose of SMT for transdermal delivery was calculated?
See, as I mentioned there earlier in that slide, So dose in the sense here based on the flux value. You see this equation, the CSS is the steady state concentration, which you get with the JSS, the steady state flux, and area of the patch divided by the clearance of the molecule. So that means using this equation, you can fit the equation to calculate the required flux. JSS is equal to CS. So you can modify this equation to get the required flux. So the required flux to get for this molecule is the 17,000 nanomoles per centimeter square using the above equation. But whereas with the maximum flux that we got with the 1.5 mm microneedle application is the 6,754, which is almost 3 times less. So like this, you can calculate the amount of the somatropton needed based on your transdermal flux into the systemic circulation, where you get the steady state concentration using the values of the CSS that you got from the literature value. It is a literature value using their infusion or the especially you get this data from the IV infusion data, or if since many molecules cannot be given IV infusion, you may get the data using either from the relative or the relative viability studies using some doses form application, where you get the CSS also. So from that data also you can calculate by taking consideration of the steady state concentration of the somatropton, and you can calculate the dose. Any molecule can be calculated like this with this equation.
Sir, here we have one more question, sir. What factors are crucial for MN length?
What
factors are crucial for MN length? MN length.
Okay. See, the microneedles are made with different materials. So the microneedles may be sturdy, or the microneedles may bend. For example, we used the stainless steel microneedles here. So and then we used the thumb force you for applying the microneedle arrays onto the skin. But there are several microneedle applicators are available, but again they are available for that specific product, but not the product that we got. And we also got that product for the microneedles we used, but we don't have that in our laboratory. So every time you apply the same pressure. So sometimes what happens is depending on the microneedle structure, it's not necessarily it's a single like a rod-like structure, a thin rod or like structure. So there are several hollow microneedles, there are bent microneedles, so spiked microneedles. So there are several microneedles using the several materials, biodegradable polymers, non-biodegradable polymers, glass materials, ceramic materials. Several people tried several types of the materials to manufacture the microneedle technology. So what happens is here, depending on the material, the sturdiness, or which in other words, the strength, the tensile strength, or the tip, the strength of the microneedle will be evaluated in the lab, and that should withstand with the pressure that you apply onto the skin using the microneedle array, so that the microneedle will not be broken or will not be into the pieces when you apply onto the skin. So this is another side of the microneedle evaluation procedure, characterization of the microneedles when you make in our lab.
Okay, sir. Thank you, sir.
I mean to say, the microneedles should not be brittle in nature. So if they are brittle in nature, they break.
Okay, sir. Thank you, sir. Once again, thank you, sir, for your wonderful presentation. Sir, thank you for spending your valuable time and sharing your knowledge to all our participants. Sir. Now,
Okay.
I would like to invite Dr. B. Pamal Reddy, Organizing Secretary, to honor the resource person, Dr. N. Buchnaidu, sir.
Thank you, madam. Thank you, Supriya, madam. So we are heartfully thankful to the Dr. Buchnaidu, sir, for providing the valuable information to all the participants. I hope all the participants was enjoyed your session. I think they will implement these ideas for their career growth. So on behalf of ACT Atal Academy and Nirmala College of Pharmacy Management, we request you to accept our token of gratitude in the form of e-certificate. Thank you, sir. Thank you very much for providing.
Okay, thank you, guys.
Thank you, sir. Thank you very much, sir.
Thank you.
Yes, sir. So, dear participants, next session will start just 11:30. Thank you, Anand.